Welcome to Jerry's Gene Scene. In the next few posts, I'm going to explain high school level Mendelian genetics. Mendel had peas. I'm going to try explain Mendelian genetics using a model phantasma. Before Mendel, genetics was viewed as a blend of features between the mom and the dad. What Mendel did different was he looked at one attribute at a time. We like to talk about this attribute or what an organism looks like as its phenotype. Let us imagine two phenotypes of phantasma. There's the pure-breeding white-eyed phantasma and the pure-breeding yellow-eyed phantasma. When we crossbreed a white-eyed and a yellow-eyed phantasma, 100%, that's all, of the offspring end up being yellow-eyed. Now if we cross these yellow-eyed offspring, we'll end up with a 3-to-1 ratio of yellow to white-eyed. Mendel would have called yellow-eyed the dominant and white-eyed the recessive. Let's see how this works. We have two different phenotypes. Therefore, there must be different genotypes. We call these different forms of the gene alleles. One allele has DNA that codes to make yellow eyes. The other allele does not make a product for eye color. So let's go back to our parents of the first cross, the pure breeding white and the pure breeding yellow, and we know that when they undergo a meiotic division to form the gametes, they're going to form two sets of gametes, but in the case of the white eye, they're both going to be white eye, and in the case of the yellow eye, they'll both be yellow-eyed. So now, when any of the gametes from the white-eyed combine with any of the gametes from the yellow-eyed, all of the offspring are going to be yellow-eyed. So now we're going to take those offspring that were just produced, we'll call those the F1 generation, and if we cross them, first of all, in a meiotic division, we'll end up with two different gametes here, one that's yellow-eyed, one that's white-eyed, and that's going to be true for both cases. Now we can see that if this gamete here combines with this one, we end up with yellow-eyed, and with this one, yellow-eyed. And if we take this one here, we get yellow-eyed, and we get white-eyed. So that was a 3-to-1 ratio of possible offspring. Keeping track of the gametes and the genotypes and the phenotypes could be quite difficult. So let us investigate an easier way of keeping track. So we'll call this one a monohybrid cross. And we're starting with the parent generation. So we'll call that the P generation. And we'll make ourselves a little chart. So we'll start with the phenotype. Then we'll go to the genotype. Then we'll go to the gametes. And finally, the offspring. And with this, we know that we're going to cross a pure breeding yellow-eyed with a pure breeding white-eyed. The genotype, we're going to choose a capital Y for the yellow because that is the dominant. And we will not use a W, we're going to use a lowercase y for the white-eyed. So we want to keep those letters the same in both cases. We don't want to get those mixed up. Now, the only possible gamete from the yellow-eyed is a big Y, and the only possible one from the white-eyed is a little y. And when those two come together, we're going to end up with a big Y and a little y, and we're going to know that that is 100% chance that the offspring will be yellow-eyed. One more set of terms just before we move on. This one and this one are each called homozygous because they are the same. This one here is called heterozygous because they are two different alleles. Now we're going to cross the F1 generation. The phenotype is they were both yellow-eyed, and the genotype of each of these, they were heterozygous, big Y, little y. So now the possible gametes for this one are either big or little, and for this one, the same thing. So here, we could have this crossing with this, or with this, and this one with this, or with this. And to make that as simpler, we're going to create what is called a Punnett square. And in the Punnett square, we make the number of squares the same as the number of gametes. So we have a big Y and we have a little y. And the same is true here, a big Y and a little y. So now this one and this one make big Y, big Y. This one and this one make big Y and little y. This one and this one make big Y and little y. Notice I bring the capital Y to the front in both cases. There's no need to have them reversed. We want to keep it so that when we look at it, we can instantly tell which color it will be. And in this one, it will be little y and little y. Now, if we look at this, this should give us a yellow-eyed. This will give us a yellow-eyed. This will give us a yellow-eyed. But the final one, this one, will be white-eyed. And that is a 3-to-1 ratio of yellow to white. And this is the F2 generation. Thanks for watching. If you want to see more about Mendel's contributions, click on the video to the left. If you want to learn more about astrobiology, check the one out on the right.